Welcome back. In this session, we will discuss other antibiotics which are not beta-lactam antibiotics and study their mechanism of action. Okay. The first thing that is there, that is of course, that we raised that point at that time, that in the cell wall biosynthesis, what happens, you require D amino acids like D-glutamic acid or D-alanine. That is very important for the cell wall biosynthesis, D-alanine. Now, the question is from where this D-alanine is coming. The D amino acid is coming from L amino acid, because in the biological system the amino acids belong to the biological system. I am not talking about plant origin, usually it is all, they are all belonging to the L configuration. So, the source of D amino acid is nothing but the L amino acid. So, in order to convert the L to D, what you need is epimerization. In organic chemistry, we call that as epimerization. Okay. And epimerization, and in epimerization, if you remember now your biochemistry, the first part of this course, that epimerization is carried out by a PLP dependent enzyme. PLP means the vitamin B6, the pro coenzyme is the vitamin B6. So, PLP dependent isomerization that takes place when you convert L to D. Okay. So, now because we do not need any D amino acid in our body, the host does not, at least to this day it is not known that there is the requirement of any D amino acid. That means, this alanine racemase is the alanine racemase is the enzyme which racemizes alanine from L to D. Okay, alanine just a second, L alanine to D alanine. This is done by alanine racemase. Okay, the enzyme will be called alanine racemase. We do not require this alanine racemase. So, if you can target the alanine racemase, that means that will be a very good target, very selective, because we do not require this enzyme. Okay. Now, what has happened, that yes, there is an inhibitor for alanine racemase, and that is what is called D-cycloserine. What is cycloserine? See, on this side you have I have written the D-alanine. This is the structure of D-alanine. What is D-cycloserine? You see, it resembles the alanine very much, that you have, instead of the methyl, you have CH2 here. The configuration is okay. This is carbonyl, instead of which you have nitrogen, and then that is connected via, this is called oxazole ring, okay, NO. So, now what will, so basically now it resembles the D-alanine. So, it can now react with the alanine racemase and then inhibit the alanine racemase, and the mechanism that is given in textbooks are like this, that you have this CO. The first step of any PLP dependent enzyme is the formation of the imine. Formation of the imine, again just to brush up that earlier thing, that this is present as a plus, and in the plus form it is an electron sink, and then what happens, this hydrogen is lost. Okay. So, this goes up to that point nitrogen, then it comes back and stays at the CH2 level. So, you have NH, O and then CH2, then you have double bond, NH, CH2 and then the pyridinium ring. Okay. So, this is the situation now. So, as soon as this becomes this plus, now the enzyme, see PLP is attached to the enzyme, do not forget the enzyme, because everything is happening inside the enzyme. So, now the enzyme nucleophile, any nucleophilic amino acid, that now, because this has a carbonyl, this is a very well known fact in organic chemistry, that if there are two carbonyls sit side by side, then one of the carbonyl is very reactive. Okay. So, the same thing is there, one is iminium, another is a carbonyl. So, this carbonyl is now very reactive. So, the enzyme any nucleophilic amino acid is going to attack here and opens this up. That means, you are forming a covalent bond, an acyl enzyme covalent bond, and that stays there. So, this is the mechanism of cycloserine, D-cycloserine. Okay. So, I think this is used in cases where it is a second line of defense for tuberculosis in tuberculosis, second line, not the first line. First line of molecules are different, isoniazide, ethambutol, rifampicin. Okay. However, if after the second line of defense includes this D-cycloserine, and the mechanism, first of all, it resembles D-alanine in structure. So, it is accepted by the enzyme, and the mechanism is that it forms the imine. Then there is basically a tautomeric shift, it forms this the imine was earlier with the aldehyde carbon, now it is with the cycloserine carbon, and then that activates this carbonyl. So, the nucleophilic amino acid attacks and opens up the cycloserine. Okay. So, that is, so it is again you can call it as a suicide inhibition, okay, because there are changes that take place in the molecule. There is another class of molecules which is quite recent, but that is entirely synthetic, antibacterial compound. They are called oxazolidinones. I think they are the one of the modern discoveries of antibacterial agents. So, oxazolidinones, oxazolidinone is this. I think this is isoxazole, this is not oxazole. Oxazole is 1,3 and this is 1,2. So, this is oxazole and that is isoxazole. So, this is isoxazole, again I repeat, isoxazole, and this is oxazole, 1,3 is oxazole. Okay. So, this is oxazolidinone, because the double bond is not there. So, oxazolidinone, that functionality along with these attachments, that can also be an antibacterial agent discovered in recent years. They inhibit protein synthesis. Protein synthesis means the translation. So, they inhibit protein synthesis. I think that is enough knowledge that they stop the synthesis of protein. And if you want to know a little bit more, that a 70S ribosome has to be formed by the combination of 30S and 50S ribosome. Again you remember that first the two subunits have to join together. Now, what happens, these oxazolidinones bind to the 50S ribosome and prevent the 30S to bind to the 50S ribosome to make the whole complex, that is the 70S ribosome. So, it stops the 30S from binding to the 50S, that is the oxazolidinone. So, that is their mechanism of action. So, it is basically it is a covalent, not non-covalent interaction. Okay. Then you have this is the vancomycin we are talking about, one of our last line of defense. Vancomycin also is it actually stops the assembly of the peptidoglycan unit. So, the peptidoglycan has to assemble, hang from the glycan units, the peptide. So, somehow this assembly, assembly means, see you have the peptidoglycans, the glycans like this, and then you have these peptides hanging from this. Okay. So, that assembly means it has to be perfectly positioned. So, that is inhibited by vancomycin. It is a big molecule, but that is again it is an antibiotic produced by a microorganism. Then you have you have another class of molecules which are called quinolone class of molecules. Quinolone, because it is a quinolone, quinoline is only 6 and another 6 with a nitrogen, but it is a quinolone, a carbonyl. So, these quinolones, they are also very good antibiotics. At some point of time they were, when they came into the market, they worked extremely well. Their mechanism of action is they target the DNA gyrase. You know what DNA gyrase does? That is actually unwinding of the double helix during the replication. Okay. So, this quinolone stops the DNA, it is an inhibitor of the DNA gyrase. That means, these are all non-covalent interactions. It binds to the DNA gyrase and the DNA gyrase or gyrase that does not do the replication, that stops the replication. One of the famous compound is ciprofloxacin. Now, this ciprofloxacin or norfloxacin, there are different generations that have come. Ciprofloxacin was the starting one, then came norfloxacin, then came levofloxacin, then gatifloxacin. Now, today, because they are becoming resistant, all are by efflux mechanisms. So, ultimately now the best drug what is coming out in the market is moxifloxacin. Let us see, moxifloxacin. This is given for urinary tract infection. Moxifloxacin works very well. Ciprofloxacin or norfloxacin now is given to prevent diarrhea. If somebody is having diarrhea, then the ciprofloxacin is given. Okay. So, we should know some of these molecules, how they work, at least in terms of, because there is no covalent interaction here. So, no arrow pushing here, that is not required. Then you have another group, tetracycline. It is a very broad spectrum antibiotic. However, the problem is, you see the problem with all these molecules, especially this quinolone, is that it is targeting DNA gyrase or gyrase, which is also present in human. Okay. So, that is why they have much more toxicity than your vancomycin or than your D-cycloserine or than the penicillins, because they target they have a different target which is not present in the host. So, now tetracycline, what tetracycline does? Remember again the protein synthesis, that you have a 50S ribosome and a 30S ribosome. You have this P site and the A site. In the P site you have this growing peptide chain, A site the tRNA comes with the particular amino acid depending on the codon here. Okay. Here what happens? The tetracycline sits at the A site and blocks the tRNA from binding there. Okay. So, it stops the protein synthesis by binding to the A site. Okay. What it says here? Okay, first of all, it is used to treat acne, respiratory tract infection, other conditions, and another important use is that when there was this problem of plague outbreak in Surat about say 10-15 years back, then the only drug that used to work was tetracycline. So, people bought this tetracycline and stored it in their home for backup. Okay. So, tetracycline is very cheap, one of the cheapest antibiotic that is available in the market. But how does it work? It binds to the A site of the ribosome and then stops the protein synthesis. Okay. Then you have chloramphenicol, another. Chloramphenicol is given in typhoid. However, again I should remind that because these molecules are targeting some system which is also present in human, so they are bound to be toxic, and that is indeed true, they are toxic. So, you have to take only the prescribed dose. Chloramphenicol is rarely used these days, but it is one of the best medicine for typhoid. And how does it work? Chloramphenicol, you see the arrow here. See, you have this A site, you have this P site, the chloramphenicol is in between here. So, what happens now? This cannot, what happens when the peptide synthesis takes place? First, this growing peptide chain is transferred to this site and then it shifts, but the chloramphenicol is sitting here, right here. So, it cannot, it does not allow the shifting possible. So, the peptide, basically the growing peptide chain cannot exit, it stops the exit pathway of the growing peptide chain. Okay. So, these are some of the other important antibiotics that I thought that you should know. Another is erythromycin. Erythromycin is also these are called macrolide antibiotics, because they have a big macrocycle. You see, they have this big macrocycle. So, they are called macrolide antibiotics. How do they work? They also inhibit protein synthesis by binding to the 23S rRNA. So, they bind to the ribosomal RNA in the 50S subunit and of the bacterial ribosome, blocking the exit of the growing peptide chain. So, they are very similar. Tetracycline binds to the A site, then chloramphenicol binds in between the A and the P site, and erythromycin, it says that it blocks the exit of the growing peptide chain. So, these are the three important ones, that how do they work. So, more or less these, there are still some more antibiotics. I think we can go to our next topic. So, again I repeat, see, chloramphenicol, then your tetracycline and erythromycin. The consequence of their action is basically protein synthesis is stopped. Protein synthesis is hindered. Okay. That is the consequence. Only thing, their binding pattern is different. Chloramphenicol binds in between the two sites, then tetracycline binds in the A site, and erythromycin binds somewhere in that, in this ribosomal subunit, which does not allow the growing peptide chain to exit. Okay. So, that is the, those are the mechanisms. Now, let us come back to more organic chemistry, and I will show you the power of organic chemistry in making newer antibiotics, and especially I will focus on the penicillin and cephalosporin, these two, because these are the still the major antibiotics that are used today. Okay. Now, remember I told you that in penicillin it is extremely difficult to substitute these methyls. See, if you can develop a method where this can be substituted with different groups, then what happens? Actually, what happens, the bacteria, when they are exposed to a penicillin, they know the structure of penicillin, they remember that. So, when the next time you give penicillin, they know that there will be a methyl here, there will be a methyl here, they immediately know that this is penicillin. But now, if you can have a method by which you remove this penicillin, this is, you remove the methyls and put some other group, suppose some other R2. So, now the penicillin, now bacteria becomes little bit perturbed, right? What is that? This is not the penicillin, same penicillin I was used to having, and I was destroying this penicillin, but now there is a new molecule. So, the bacteria may be killed by the process. Okay. Before it can realize what is coming here, it can be killed. Okay. However, to make different R's here is extremely difficult, but possible, and I will show you how it has been done. But in order to go into that details, what we need is the question, the very basic question, that two questions: why bacteria makes penicillin? Why does bacteria or fungus, whatever, or fungi make penicillin. Okay. I think I gave you the answer here, because like us, the bacteria also is constantly bombarded by or constantly attacked by other bacteria. So, it has to make some molecules to kill the surrounding bacteria. So, that is why one of the defense, these are defense molecules, this is their weapons basically, penicillin, cephalosporin. So, they are their weapons. Okay. That is number one. Number two is, how does it make the penicillin? How does it make? What are the starting points? What are the building blocks? Now, that is shown here. I told you when the penicillin was introduced to you that there is this penicillin is made by a very simple sequence of steps. One is that there are 3 amino acids, this is L-cysteine, this is L-valine and this is L2-amino adipoic acid or adipic acid, 2-amino adipic acid. Okay. So, these 3 amino acids combine and to form a peptide. Okay. Amino acids combining through carboxy and amine means they are forming peptide bonds. So, 3 amino acids combined means you get a tripeptide. So, this will be the tripeptide that you get. Now, there are few questions here. See the configuration of the valine here, L carboxy is up, hydrogen is down, and when it makes the tripeptide, what is the configuration here? Now, it is D, the carboxy down, hydrogen up. Okay. So, this molecule while making this tripeptide, there must be an enzyme which is doing some racemization here, L to D, but when people thought that instead of L-valine, I give D-valine, so that the bacteria accepts the D-valine and makes this tripeptide, but it does not accept D-valine. It will only accept L-valine and then it will epimerize it into the D. Okay. So, that is one interesting aspect. The other is that this 2-amino adipic acid or alpha amino adipic acid, it is the side chain carboxy which participates in the peptide bond formation. Okay. So, now you have this alpha amino adipoyl cysteine valine and that is called L, abbreviated as LLDACP. L for this A, L for this C cysteine and D for V. Okay. Full name is L alpha amino adipoyl L cysteine D valine. Okay. But you cannot write all the time this big name. So, LLDACV. Now, this LLDACV, once the peptide is formed, the tripeptide, now there is an enzyme which is called isopenicillin N synthase, isopenicillin N synthase. Synthase means it does not require any ATP to make the molecule. Okay. What it does? It first forms this nitrogen carbon bond and it forms this sulfur carbon bond, and in one step you get this molecule. Now, this is what is called isopenicillin N, not penicillin N, because earlier this molecule was discovered where this is having a D configuration and that was named penicillin N. So, the scientists have no other option to call it as isopenicillin N. So, penicillin N was known earlier, but then they got this L configured alpha amino adipoyl system. So, they had to call it isopenicillin N, and that is why the enzyme which makes the penicillin from this tripeptide is called isopenicillin N synthase, because this is known as isopenicillin N. Okay. And now, in some organisms it stops here, but some organisms what happens, this is further converted into penicillin N, that means an epimerization has taken place, and then another enzyme comes and converts this penicillin into cephalosporin. So, that means penicillin and cephalosporins are two sisters or brothers, whatever you say, and then they belong to the same class and they start from the same molecule. So, that is the tripeptide which is formed from 3 amino acids. Only interesting is that valine, L valine gets isomerized to D valine. So, LLDACV, if you add IPNS, that isopenicillin N synthase, then what happens? It is oxidized. This is nothing but an oxidation, a double oxidation, nitrogen carbon bond formation and sulfur carbon bond formation, and that ultimately goes to isopenicillin N. You can just for the time being, we just exclude this part, that how the cephalosporin is obtained, but just to know that cephalosporin is just a further downstream process. From penicillin you can get cephalosporin, and that what happens in microorganisms. Okay. Now, this very interesting chemistry. So, whatever we are saying is that first these 3 amino acids combined to form this tripeptide methyl SH, that is cysteine, and you have NH, and this is the adipoyl side chain. I am not writing that. Okay. So, there is an enzyme called IPNS, and that makes what? Remember what we are studying now? The biosynthesis of penicillin. How penicillins are made in the microorganisms, that is what is called biosynthesis. Like if you say how Taxol is made in the plant, that is biosynthesis of Taxol. So, this is biosynthesis of penicillin. So, this enzyme, one enzyme, just look at this, one enzyme does 2 reactions at the same time. One is this formation of this carbon nitrogen bond, another is formation of this carbon sulfur bond. And while doing that, you have to remove this hydrogen, and you have to remove one hydrogen from here and the NH hydrogen from here. So, that means 4 hydrogens are removed. So, what is the fate of the hydrogens? The fate of the hydrogens is 2 molecules of water. So, from where the oxygen comes? Actually, this reaction requires oxygen. This reaction requires iron as a cofactor, ferrous, and you know any ferrous requiring enzymes need vitamin C, the ascorbate, to keep the ferrous, to keep the iron in the +2 oxidation state. Otherwise, if it is there is no ascorbate, it will go to the ferric. So, these are the things that requires. That means, oxygen is a co-substrate that is converted to water, IPNS is the enzyme, ferrous is the cofactor, okay, and ascorbate's function is to keep the ferrous iron in the +2 state. So, this is the reaction. The question is now there is first question that was asked is, see why I am studying this, because now if this tripeptide is accepted as the substrate for IPNS, now if you take another tripeptide, suppose this, because tripeptides are easy to make, NH, CO, and then you put some group, suppose I put a allyl group here, change that to methyls, put an allyl group. The question is, if this is accepted as a substrate, then I get a new penicillin which is entirely different from the penicillin that bacteria used to see, right. So, you will get a vinyl penicillin now. Similarly, if you can see, there are, this is the work carried out at by Sir Jack Baldwin. He made various penicillins. He first showed that IPNS can be used in this fashion, and I think he showed that he in his lab at least 100 new penicillin molecules have been made. But these are, remember, these are penicillins where the skeleton, the pharmacophore is changed basically. You are replacing the methyls now. That is synthetically is not possible, very difficult. So, if you take this allynic amino acid, now you get this allynyl penicillin. Okay. But this was possible only when the biosynthesis was known, this enzyme was characterized. Okay. Now, so basically why do you study biosynthesis? Because that is one of the way you can, that will help the drug discovery processes. Okay. And from organic chemistry point of view, they are extremely interesting. Like if I now ask you that how many bonds are formed in this reaction? 2 bonds. One is this carbon nitrogen bond, another is the carbon sulfur bond, this carbon and sulfur. Which bond is formed first? Mechanistic organic chemistry, the first question is which bond is formed first? Because if this carbon sulfur bond is formed first, that means you get a macrocycle in the beginning like this, and then the carbon nitrogen bond is formed. The other is that carbon nitrogen bond is formed first, that means question is which is the intermediate for this reaction. If this is the intermediate, if this is the intermediate, that means carbon nitrogen bond is formed first. On the other hand, if you get this, then you can say that the carbon sulfur bond is formed first. So, people tried to see which intermediate is formed, but they could not find any intermediate. Okay. Then they did a reversal of their strategy. What they did? They made this compound, this macrocyclic compound, and give it to the enzyme and see whether the enzyme converts it into the penicillin or not. And what happened? This was not converted into the penicillin. Now, the question is then it pointed out that possibly then this is the intermediate that is formed first. So, they wanted to make this molecule and showed it to the enzyme, and they thought that they will immediately get the penicillin. However, the problem was this molecule has a very short half life. Within 15 minutes, it is destroyed. Why? Because you have a beta-lactam, you cannot have a beta-lactam where there is a XH here, because immediately this will happen, the ring opening reaction. So, this thiol nobody could make. So, that means nobody could demonstrate that this is the one which is the intermediate. That means nobody could demonstrate it directly that the carbon nitrogen bond is formed first. See, negative, this is a negative experiment, that this is not converted into penicillin, this is stable. So, that is not a direct proof, you cannot just say immediately that means that the carbon nitrogen bond is formed first, that is not, because that is a negative way of saying this. You have to show it that the carbon nitrogen bond is formed first. How do you do that? A very simple experiment that was done. I think we will stop after this. You take, this is an indirect experiment, because direct you cannot do it, because you cannot make that intermediate. So, this compound, this tripeptide was made, where all the hydrogen, all the positions are occupied by the lower isotope, that means the hydrogen, the normal hydrogen. Then this molecule was also made with proper stereochemistry, 2 deuterium Cr, okay, and then NH COR. And the third molecule was made, SH, which had a deuterium at this position. Okay. Suppose this is A molecule, this is B molecule and this is C molecule, and then A and B are mixed together. So, now you have two test tubes. Here it is A plus B. Suppose just for assumption that you suppose think that this is 1:1, and in another test tube you mix A plus C, that is also 1:1. Okay. Now, what you do, you add your IPNS, you add the iron, whatever requirements are there, ascorbate, all these things. Okay. So, the reaction started because you have given the tripeptide. So, reaction will form penicillin. So, experiment what was done? They took some aliquot from here, a little aliquot, put it into the mass spec. So, what will happen in the mass spec? Because say after 5 minutes, so there will be lot of A and B still present. So, what will happen now? You will see two peaks, one for A and one for B. What is the difference between A and B? 2 mass units. So, this is M, this is M plus 2. Okay. And what you do for the other test tube? You do the same thing, start the reaction and check the mass spec, that means check the ratio of B and C. Okay. B and C, check the ratio and then see whether the ratio changes in any of these. Very interesting experiment, because it contains physical chemistry also now. So, you get M and M plus 1, because it has got only one deuterium. Now, what happened? What was the result? The result was that or I do not show the result first, let us first analyze that suppose this bond is formed first. Okay. So, when there is A and B, then what will happen? There is this deuterium here. You know the deuterium loss is more difficult, you get a huge isotope effect. So, because if this bond is formed first, so initially what will happen? The enzyme will pick up this and not the other one, preferentially pick up this and not the other one. So, if that be the case, that means A and B ratio will change, and if this bond is formed first, the carbon, the sulfur carbon, then there will be no isotope effect here. So, both will be transformed into the intermediate, and then later on when it finds deuterium, it can throw it off, that I will do that later on. But what happens? This will be processed to the intermediate, and that will be processed at the same rate to the intermediate, provided the carbon sulfur bond is formed first, because there is no isotope effect here, because this is also hydrogen, that is also hydrogen. Okay. On the other hand, if you take A and C, what will happen? If this bond is formed first, then both will be accepted as a substrate with equal preference, but the second step will be a problem, carbon sulfur. Okay. So, the conclusion is that if this is the first step that is formed, then A is to B is going to change, and if this is the bond that is formed first, then A is to C is going to change, because that means you have to see where is the isotope effect. Isotope effect will be shown in the first step, because that will give you the ratio of A and B. Basically, it is energetically it is like this. See, if it is A and B, if there is no isotope effect here, that means if there is no deuterium, then all A and B will come here, but those are not A and B, those are transformed A and B. Okay. So, but if A and B are processed not at the same rate, then there will be accumulation of one of these, especially the deuterated ones. What was the experimental result? The experimental result was that the experimental result was that the ratio changed here, but on this side the ratio remained the same. So, what does it prove? If the ratio changed here, that means A and B, the first step you phase face the isotope effect. So, first step you say you face the isotope effect only when this carbon nitrogen bond is formed first. If it is the second, then A is to C should have changed. This is actually A, sorry, I am very sorry, that is A, that is not B. Okay. That is AC and that is AB. So, this got changed. As the time goes on, you will see that even it is less. B will be only processed when all A's are consumed, then only the B will be taken up. Okay. So, you have to study this at initial points, that what is the ratio of A is to B and what is the ratio of A is to C, and that experiment was done and it was proved that this is the one which is the rate determinant, which is the first step in the process of making the penicillin. Interestingly, the question can be asked that if the enzyme can make this and make it stabilized and then form the next reaction, chemically when we make in the solution, this does not stay, okay, because the enzyme has some way to stabilize this thiol. Okay. I told you that some people tried to make this compound and give it to the enzyme, but they never succeeded in that because it has got a very poor half life, especially at pH 7.2 where this IPNS works, it does not stay for a minute even. Okay. Only at lower pH 20 minutes is the half life. Okay. I think so that we can in the next class we will discuss the mechanism of formation of this, that means the cofactor chemistry, that is I think that will take more time. So, we will consider the biosynthetic mechanism for penicillin formation as well as erythromycin formation, and we will in the next class we will also show how you can make different types of penicillin, different types of erythromycin by what is now called the genetic engineering or protein engineering. Okay. Thank you.